Hey guys, how are you doing? Today in this video, we are going to talk about CAR T cell therapy, a revolution in cancer treatment using your own immune cells to fight cancerous tumor. But why does this miracle cure cost over 40 lakhs in India? What makes it so complex? Let's break down the science, challenges and UPSC relevance. Before we begin our video, let's go through the recent newspaper headlines, and this was in the Hindu on 21st June 2025, and it says you don't need to take your T cells and grow in the lab. Instead of extracting T cells and engineering them in a lab, they are saying researchers have delivered messenger RNA directly into circulating immune cells, right? That is one newspaper headlines. You might have come across newspaper headlines like engineering medicines, the future of CAR T and CRISPR technology. This is very important for your examination. CRISPR system, a gene editing system. They can ask you question about it. Again, another news headlines reads as the following: a cutting-edge cancer therapy offers hope for patient with lupus. Lupus is basically autoimmune disease, right? It is autoimmune disease, right? Okay, and a treatment called CAR T appears to stop it in its track. So let's start. Let's talk about, and obviously before getting into these discussion, you should be writing these answers after this video, and some of these questions for you. What is CAR T cell therapy? Discuss its underlying scientific principles and evaluate its potential in cancer treatment. Directly science and tech type का question है. You could also write another question. CAR T therapy holds promise for treating certain cancers. These days they are not only talking about blood cancer but solid cancers also. Treatment of solid cancers through CAR T cell therapy. Okay, you should mention all these things. But remains unaffordable for most in India. You should talk why there is unaffordability, why it is so expensive. Analyze the challenges and suggest policy measures to make such therapies accessible. Okay, इस टाइप को आपका आंसर होना चाहिए. Another question for you, practice question. Innovative therapies like CAR T have widened the healthcare access gap. Discuss the ethical and regulatory challenges in ensuring equitable access to life-saving biotech innovations in India. Okay, this is again about biotechnology. Another question. This question reads as the following: examine the role of public-private partnerships and regulatory reforms in scaling advanced therapies like CAR T in India's biotech sector. Okay, this lecture will not be obviously suffice for it. It will give you the glimpse, the basic idea, the core for this CAR T cell therapy. You should be reading newspaper headlines. You should be following them, and then you will be in a position to write better answer. Okay, or maybe best answer maybe, right? What is CAR T cell therapy? This stands for basically chimeric. This stands for chimeric antigen receptor. Okay. Before we get into this system, this CAR T cell therapy, let me tell you some basic idea. What is the foundation of our immune system? We have discussed it in another video, and you should go through that video again if you have not once gone. What is the foundation? What is the concept? Let me tell you. WBC. White blood cells they make up the defense system for our body, right? They make up the defense system of our body. There are two types of cells, two types of WBC basically, and it is also called leukocytes. They are called leukocytes. One is called granulol. One division is granulocytes. Granulocytes mean they have granular structure. Another division is agranulocytes. Agranulocytes mean they have structures which is not granular shaped, right? They are of three types. One is called neutrophils, right? Neutrophils are essentially they fight against infection, right? They make up 60% of WBC, and the WBC count should be around 5,000 to 7,000, or maybe if you read for that matter, if you read some blood report, you will learn that it is around 7,000 to 10,000 per microliter or per mm cube, right? Just remember, this is WBC count. And if you were to take 10,000 as a base number, then 6,000 there should be neutrophils. That is essential idea. And remaining 30%, around 30% are lymphocytes. So they make up together 90%, right? Then there are eosinophils, right? EOSINO, eosinophils, right? Then there are basophils. Eosinophils, they fight against some kind of, you can say, drugs, right? Detoxification basically, right? And the basic function of basophils is to fight against allergy. Lymphocytes are, the topic of our discussion today. Lymphocytes, they have primarily two type of cells, right? Let me tell you. One is called B cells, other is called, and maybe I should write first T cells, which is our discussion. T cells, and others are called B cells. In one word, let me discuss this. They are of two types, lymphocytes, and others are called monocytes. Okay, I am not going to talk about monocyte. They are highly motile and they are phagocytic in nature. T cells, they have cell-mediated immunity. Cell-mediated immunity. What they do actually, they produce some kind of toxic chemicals and kills pathogens, kills antigens basically. B cells, they are called antibody mediated. Antibodies are proteins, antibody mediated humoral, also it is called humoral, immunity, mediated immunity. Okay, so these cells are responsible for antibodies. They produce antibodies. T cells, they are cell mediated immunity. They are phagocytic in nature. They produce cytotoxic, chemicals or toxic chemicals to kill, pathogens. Now, let me tell you. So, this, those WBC are, WBC typically they have. Let me take this one. This amoeba type shape, है ना? अमीबा की तरह ये अपना शेप क्या करते हैं, चेंज करते रहते हैं. Therefore they are called as amoebic shape, है ना? They have amoebic shape. Now these amoebic shape has some kind of proteins are there on their surface. Some proteins are attached to their surface, right? These protein receptors are cow-like. These protein receptors have claw-like. What they have actually? They have claw-like structures, है ना? पंजा होता है जैसा, claw-like structures. And ये पंजा क्या करेगा? ये claw-like structure करेगा. ये किसी फॉरेन मटेरियल पर, मान लो कोई कैंसरस सेल है, सपोज यहां पर कोई कैंसरस सेल है ऐसी, ये इस पर क्या हो जाएगा? चिपक जाएगा. इट विल लैच ऑन टू एंटीजेंस, राइट? तो क्लॉ लाइक स्ट्रक्चर, दे लैच ऑन टू एंटीजेंस. जैसे ताला चाबी होता है ना, की एंड लॉक सिस्टम, इस तरह से उसको क्या करता है? डिस्ट्रॉय करता है. एंड व्हाट इट डज नाउ, इट विल रिलीज. नाउ वंस इट इज अटैच्ड टू इट, इट विल रिलीज. Let me write down here, okay? Now, T cells, what they do? Now T cells release toxic chemicals and destroy. Release toxic chemicals and destroy it. B cells and T cells, they work together, and if they find any kind of infection in the body, what they do actually, they expand in the numbers. For example, limbs. There are lymph nodes, for example, there is, lymphs in case of sore throat, we find, kind of expansion. थोड़ा सा यहाँ पे ऐसा हो जाता है, sore throat में बढ़ जाता है. What is happening actually? Number of B cells and T cells are increasing in numbers here, and they work together. They find a common target. Once they keep on exchanging, they interact billions times in single minute, right? And B cells have ability, T cells have ability, B and T cells have ability to, identify rogue targets. Okay, but sometimes what happens, these cancer cells, there is one thing actually. What happens, sometimes cancer cells, let me write it somewhere or maybe here, sometimes cancer cell fool T cells. Okay, sometimes what it does, cancer cell fools T cells by developing ways, and what are the ways? Typically two ways. Either disguising healthy cells, or what they do actually, they are sprouting so many antigens, on their body, जिसके कारण क्या होगा? T cells will not have a hold on, those antigens, and they will, escape, this cytotoxic mechanism, right? Now, what is happening now? This CAR T therapy, let me tell you. Before how it works, let me tell you basic idea about this CAR T cell therapy. Okay, just a minute. ओके, तो एक बार मैं इस स्लाइड को आपके पास फिर से लेके आ जाऊं। ओके, तो ये मेरे पास था, है ना, तो इसने फुल डाउन कर दिया। तो कार्टी सेल में क्या होता है बेसिकली, दिस chimeric antigen receptors, it is the process, it is based on the retraining of what it does, retraining T cells, retraining T cells to recognize some kind of tag, है ना, to recognize and destroy rogue targets, RO, ठीक है, तो let me GUE, rogue targets, ठीक है, rogue targets. This is what is the concept of this chimeric antigen receptor. Okay, they are essentially in one line reprogrammed T cells. सरल भाषा में हम इनको बोलेंगे तो what are they actually? Reprogrammed T cells. You have changed their gene, reprogrammed T cells, right? So they have, like, as earlier I was talking, suppose this is now WBC as they are nucleated cells. They have some kind of proteins on their surface, already present proteins. Now this chimeric antigens have specific tags, it shows some kind of, these are synthetic, CAR, chimeric antigen receptors, and they will latch onto, they will find a tag, what they do actually, they will find a tag on rogue targets, and then release, again unka mechanism same hai, release cytotoxic chemicals and kill those, kill or destroy. This is what is the mechanism. लेकिन अब इसका प्रोसीजर क्या होगा, दैट इज मोस्ट इंपोर्टेंट थिंग, राइट? द प्रोसीजर इज ऑलमोस्ट सेम एज इफ इट इज you are going for blood donation, okay? And how does it work? The patient, what it does actually, T cells are collected from patients, like what? Blood donation camp. It is similar as the blood donation procedure, if not camp, right? तो blood donation का जो procedure होता है same to same, उसमें क्या करते हैं हम लोग, T cells को क्या करते हैं, collect करते हैं, and the next stage is these T cells are genetically engineered. It simply means a gene is introduced, right? Gene is introduced. How it is done? This is done typically with the help of a virus, and which should be a modified one, or it is inactivate, है ना, inactive virus. तो inactive virus की मदद से what we do actually, we introduce a gene, and this gene it encodes for, what gene do actually, they encodes for a protein. It encodes for a protein, and it produces CAR, this chimeric antigen receptors. Okay, this is what it does, right? So CAR T cells are, now next is what? These cells are able to recognize and kill cancerous cells to help guard against recurrence of this event, right? These re-engineering cells are multiplied, right? And typically it takes 7 to 10 days, and then in a stepwise manner, after 21 days, they are slowly, small doses are given, small doses are given. So they are first multiplied, and thereafter they are infused back. T cells are infused back into the patients. Okay, similar manner में जैसे हम क्या करते हैं, blood चढ़ाते हैं, है ना? Inside the body, CAR attaches to antigen on the surface of a cancer cells, because it is now expressing, these CAR receptors, and these CAR receptors are able or recognize typical tag. For example, the ability to detect, is typical tag. Example of this typical tag is, for example, CD19, and this is expresses almost in all major, B cells, right? Okay, those are culprits of cancer basically, all right. These reprogrammed cells now release powerful cytotoxins causing cell death, and this is what is the technique, all right. So I hope you have understood the basic understanding. So when T cells fails to identify cancer cells, we scientists have discovered a way to enter a gene, through genetic engineering, and that gene encodes for a protein which makes CAR receptors. That's the basic idea, right? So, but what are the challenges? First challenge is what? This process is very complex. Therefore, this is very complex process. And this complex process, because of two things, it is very expensive, or you can write for slow. This process is very slow, and it is slow, and therefore it is expensive also, not only for slow but complete, advanced, very sophisticated. First thing first, it requires lymphodepleting chemotherapy. What is lymphodepleting? I think you have understood lymphodepleting. Okay, lymphodepleting chemotherapy, this is done so as to make space. भाई आपको नई टी सेल्स देनी है ना, तो उसके लिए क्या करना पड़ेगी, जगह बनानी पड़ेगी, है ना। तो प्रेपरेटरी ट्रीटमेंट बेसिकली, व्हाट इज दिस? दिस इज अ प्रेपरेटरी ट्रीटमेंट, एंड दिस प्रेपरेटरी ट्रीटमेंट इज डन टू वाइप आउट patients existing T cells, right? Okay, so I am using pen tab, maybe this handwriting is legible. I think you should be able to read it. Okay, existing T cells to make space for modified T cells or simply for CAR T cells, है ना। तो पुरानी सेल्स को जब रिमूव करोगे तब जाके CAR T cells के लिए क्या बनेगी, जगह बनेगी, राइट? यू गेट स्पेस। नाउ बट व्हाट इज द प्रॉब्लम एक्चुअली? कॉमन सेंस अगर आप लगाओगे, इफ यू यूज कॉमन सेंस, once a person is deprived of his own immune cells, it can have risk of serious secondary infection, right? बॉडी में जैसे HIV एड्स में होता है, हमारा इम्यून सिस्टम वीक होता है, वहां पे तो इम्यूनिटी वीक होती है, बट यहां पर हमने क्या कर दिया, इम्यून सेल्स को रिमूव कर दिया है। तो दिस इज रिस्क ऑफ सीरियस सेकेंडरी इन्फेक्शन। व्हाई? ड्यू टू लो immunoglobulin level, right? Because of this low immunoglobulin level, there is risk of serious secondary diseases. I hope you have understood it, okay? And इसके अलावा एक दूसरी प्रॉब्लम है रिस्क के अलावा, it necessitates prolonged and recurrent hospital admissions or hospital visits at least. Okay, बार-बार हॉस्पिटल जाना पड़ेगा, hospital admissions. Okay, these are the serious problems, right? Okay, another challenge is what? Why it is so expensive? Why this treatment is so expensive? Simple logic. Okay, जैसे इसमें क्या होगा, जो it is kind of personalized cell harvesting, right? तो personalized cell harvesting है ना, अगर मेरी सेल्स हैं तो मेरी सेल्स को हार्वेस्ट किया जा रहा है केवल। ओके, एंड वी आर गोइंग टू टॉक अबाउट व्हाट साइंस एंड टेक्नोलॉजी हैज बीन इमर्जिंग ओवर अ पीरियड ऑफ टाइम। पर फिलहाल तो क्या है कि पर्सनल सेल हार्वेस्टिंग है, इसका मतलब है 30 से 35 लाख। इट कॉस्ट अराउंड 30 to 35 lakhs, right? To manufacture, in easy words, to manufacture individuals T cells, है ना। बहुत कॉम्प्लिकेटेड प्रोसेस है ना। इट टेक्स सो मच सेफ्टी प्रोसीजर। ऐसा ना हो कि मान लो मेरी टी सेल्स को ग्रो किया जा रहा है एंड एट द सेम टाइम देयर इज सम सॉर्ट ऑफ इन्फेक्शन गोइंग ऑन, है ना, कोई वायरल इन्फेक्शन हो जाए, कोई फंगल इन्फेक्शन हो जाए, तो फिर तो मौत क्या हो जाएगी, निश्चित हो जाएगी, है ना। तो यू शुड अंडरस्टैंड व्हाई इट शुड बी कॉस्टली अफेयर, राइट? एंड देन रिमेनिंग 30 to 35% goes for hospitalization, है ना। तो remaining, for that matter, depending upon the 10 to 20 lakhs, है ना, it goes to hospitalization, right? Not only for hospitalization, for secondary infection also. Hospitalization, right? Side effects, for side effects also, right? It goes for side effects. Post infection care also, right? Post infection care also. So I hope you have understood why it is so costly, right? What government India is doing, let me tell you. So government of India has now launched a mission called Next CAR-19. Next generation. This is about the next generation CAR T cell therapy, in which researchers are devising ways to overcome the limitation of CAR T cell therapy, right? First indigenously developed CAR T cell therapy. Another thing जो मैं आपको बताना चाह रहा था, one CAR T therapy for blood cancers. मतलब researchers have developed a potential universal immunotherapy for blood cancers like leukemia and lymphoma. मतलब जैसे ही हम blood donation के लिए जाते हैं या blood लेते हैं, in a similar fashion, in a similar way, one day will come, you know, these CAR T cell therapy will be taken like, you know, like we go for blood in a blood bank, right? So this is the way, right? So this is Government of India is doing, designing the affordable and accessible next CAR-19 marks a critical step towards self-reliance in oncology care, right? Reducing dependence or expensive imported therapies and strengthening India's position at advanced cancer treatment. Okay, and treatment and biotechnology research. Biotechnology is very important for UPSC, है ना? तो आपको पता होने चाहिए कि कौन कौन से areas. This is in your mains syllabus. Biotechnology word is given there. Okay, so they can ask you this question. So thank you so very much. What I expect from you guys now, before winding up this video, you should be writing answer to these questions now. And like, you know, like this question, policy measures to make such therapies accessible, like we were talking about next generation. CAR T cell therapy, regulatory and ethical challenges you should be writing, and biotech innovations, as I was saying, right? So now we wind up this video. Thank you so very much. Goodbye. See you guys until next time. Till then, thank you very much.